Hi, this is Shelley Kraft, and welcome to SNNLive. We're on location at the William E. Hanna Jr. Innovation Center at Shady Grove here in Rockville, Maryland. Standing with me is Jeff Galvin of American Gene Technologies, a private company. Jeff, welcome to SNNLive.
Thank you, Shelley. Good to be here.
It's good to have you. Let's get right into American Gene.
Well, we're a preclinical company. We're working on viral vectors to cure cancer.
Simply said, tell us a little bit more information, and how are you different from other companies?
Sure. So we are working with two different types of gene expression systems. So our expertise is in actually carrying therapeutic genes into humans in the clinic. And we have a couple of different drugs that are in development in the preclinical stage. One is for prostate cancer, and the other one is for liver cancer. So we found therapeutic genes that significantly mitigate cancer in vivo.
That's big stuff.
It is. It's obviously a huge market. There's one of with the liver cancer indication. It's an orphan indication, so less than 100,000 people get that every year. There's pretty much no cure for late-stage liver cancer, as you're probably aware of. And overall, the market might be in the several billions of dollars, and we're talking about stuff that we're doing in the lab that looks like it could knock it back for years. So we're not talking about something that might increase survival by a couple of months. We're talking about maybe 2 to 5 years.
What's your background? And please tell the audience.
Well, my background is actually in computer science. So my degree is in economics and computer science, and I worked in Silicon Valley for a long time and had a good success there with a variety of startup companies. And I was retired, and I heard about viral vectors from somebody at NIH. I thought this was very interesting because viral vectors sort of treat the human cell like a computer. We can go in and we can actually reprogram the DNA in a cell, and we can, in a targeted way, sever gene pathways that are essential in cancer but not important to the rest of your body. So in other words, we can make therapeutics now that are so specific that they only affect cancer cells in the body. I thought this was an unbelievable opportunity, and I thought it also related to my background in computer science, because a lot of the logic that we apply is similar, where we think about what is the condition we're looking for in the cell, and that condition turns on a therapeutic gene. That gene severs an essential pathway, and the cell naturally dies. So we're looking at a future of cancer treatment that would not be toxic, like chemotherapeutic or treating it with radiation or something like that, like all the traditional therapies. So we focused on liver cancer, because that's something that is just wide open for us. It means that the regulatory process and the cost is lower. Getting into the clinical trials when there's no other alternative for these people that are dying perhaps in as short a time frame as 6 months means that the FDA will put it on a fast track. That lowers our cost overall. It makes it a practical therapeutic for a company our size to pursue.
Where did the therapeutic, who developed that?
We developed it in our own lab, actually. We tested a number of different genes that we thought would be toxic to liver cancer, and we actually had some interesting discoveries, and now we have a combination of genes that we've put in a single vector that is showing really great promise in the lab.
You know, I think it's very interesting that you have a computer science background, as it seems that a lot of the new think tanks are taking scientists from different, interests and bringing them into biotech, and it seems to have an amazing reward for the development of both diagnostics and therapeutics. So it's interesting to hear that that's your background. Where do you go from here?
Well, we've got a bunch of NIH funding to use our technology in a couple of different indications, and that's propelling our research in the lab. And we're also looking for outside investors now to take our liver cancer therapeutic into human trials. And we believe that in the next 3 years, we'll get results in humans that'll prove that this is a practical way to treat liver cancer.
So you're probably pretty close to wanting to talk to some partners.
Of course. Yeah, well, we're always out talking to major pharma companies and mid-cap pharma companies as well. And I'll tell you that, I guess if I was a big pharma company and I controlled the market for cancer drugs, I wouldn't really be interested in funding something that might obsolete billions of dollars of my revenue. So maybe that's not the best, most fertile ground right now. But at some point, this is going to go and reach critical mass, and we're already seeing gene therapeutics come on the market. Actually, last week, the EU certified the first gene therapeutic in Europe. So this is something that even big pharma, big news. Even big pharma companies are not going to be able to ignore this market much longer. So you could see that they're resistant to really spending a lot of money on stuff where they're already making revenue on those indications. However, their patents will be running out, and they will be looking for new things. And then also, the efficacy of this will be undeniable. When you're looking at something like, and if you don't mind me, saying a brand name, Avastin, for instance, there's a lot of places where it's used where it might extend, the average extension of life might be a few months for something like $100,000. That's not really economically efficient, although, of course, that's really great.
Similarly, in prostate, I have seen the same thing.
Yeah, that's absolutely the case. So we need therapeutics that have higher efficacy, and gene therapeutics are clearly going to be a place where this is going to happen. Now, we have a lead over other labs, and we're on the cutting edge, even as far as university labs go, in terms of delivering multiple genes in high expression into human cells. And that's the kind of strength that we'll bring to this market and that will help us to develop these therapeutics.
Why Maryland?
Well, Maryland's an excellent place for biotech. First of all, think about the proximity to NIH. I mean, this really is...
And FDA.
And FDA and everything. But to me, because we're early stage, NIH is a place that really understands what we're doing. That's why we've gotten good funding from them. And of course, the next thing we need to do is work with the FDA, as you pointed out. And, there's this high-tech corridor with lots of talented scientists and engineers, and so we're never short on applications when we have a job open. And, the funding environment's pretty good around here as well.
Ladies and gentlemen, what we have here is American Gene Technologies, Mr. Jeff Galvin. It's a private company. For more information, let's get that website out there.
It's agtii.com or AmericanGeneTechnologies.com.
We are at the William E. Hanna Jr. Innovation Center at Shady Grove in Rockville, Maryland, on location with SNNLive. Thank you for coming on to SNNLive.
Thank you, Shelley.
Good to have you.